Alright, so let's talk more about how do antibiotics work. Specifically, let's talk about how the penicillin works. So what happens here is that the penicillin goes in the bacterial cell wall and it binds to the peptidoglycan. And essentially what is happening here is that it's making a hole in the cell wall. And because the bacteria is living in water, what happens is the water goes inside the bacteria and it generates pressure. And so eventually what happens is that bacterial cell gets bigger, bigger, and bigger, and eventually there's like enough pressure that the bacteria explodes. And a typical example that I use in my class is that imagine that my hand is the bacteria. And if I put a bag on top of my hand, and then I put my hand in water, is my hand gonna get wet? No, it's not, right? Because you have a bag that is protecting it. So the bag represents the cell wall. But now imagine that I'm making holes into the bag. So that's basically what penicillin is doing. So I'm making holes into the bag. Now if I put my hand into the water, is my hand going to get wet? And the answer to that, in this case, it is, right? Because now the water is going to be able to go to the holes that I made through the bag. And that is essentially what penicillin does. It weakens the cell wall, or it makes holes, if you will, so that way actually water can go inside, and because there's enough pressure, then the bacteria explodes. And as we discussed in the previous chapter, water is a very powerful and it's a very special molecule. It is so special that we actually even have a specific term that we use in order to define the movement of water. Osmosis is the movement of water across a membrane. And so the movement of water goes for an area where there's like less particles to an area where there is high particles. And that's because water likes to dissolve things. Water likes to destroy things. So it's gonna go where there's more of these particles that needs to be destroyed. So let's talk a little bit about some terminology. And this is a lot of times very confusing to people. And so what I'm gonna do is I'm actually gonna do a video. I'm gonna try to do a video where I'm gonna show you guys different solutions. So in a hypotonic solution, okay, it basically means that imagine that I have a beaker, and into that beaker, I'm actually going to place a cell. And in this case, if we're talking about a hypotonic solution, then that means that there's more particles inside the cell. There's more salt, proteins, or whatever you want to call it. So there's more of these solids inside the cell. And because water likes to destroy things, what's going to happen is that the water is going to go inside the cell. And because the cell is acquiring more water, then what's going to happen to the cell is that it's going to get bigger and bigger. Okay? And if there is enough pressure, then what's going to happen to the cell is that it's going to explode. Okay? In an isotonic solution, there is an equal amount of solutes inside the cell and outside the cell. So does that mean that there's nothing happening? No, there is stuff happening. There's actually water coming inside the cell, but there's also an equal amount of water going outside the cell. So nothing happens essentially to the cell, if you will. And the last type of solution, it's a hypertonic solution. And so what happens here is that there's more solutes, so there's more salt or there's more proteins outside the cell than inside the cell. And if you guys remember, water likes to destroy things. So water is going to go to where there's more of these particles that needs to be destroyed. So in this particular case, in a hypertonic solution, water goes outside the cell, and what happens to the cell itself is that it becomes smaller, or it shrivels, if you will, or shrinks. All right, so there are two main types of bacteria, gram-positive and gram-negative bacteria. So what's the difference between the two types? So we in the lab, what we do is that we use a dye to label the bacteria, to label essentially the cell wall. So what happens is that this dye binds into the cell wall and it turns the bacteria kind of like purplish, dark color. So if it binds into the cell wall and it turns the bacteria purple, then we call that a gram-positive bacteria. If, however, this dye doesn't label the bacteria, then we call a gram-negative bacteria. bacteria. And the reason why it doesn't turn the bacteria kind of like dark color is because it has an extra layer of fat and it prevents the dye from actually binding into the cell wall. But why does it matter if you have gram-positive or gram-negative bacteria? Well, it matters because we need to know what type of antibiotic you should be prescribed. And the reason for that is because if you give the patient the wrong antibiotic, that's a good way of making the bacteria become resistant to the antibiotics. Now, besides penicillin, there is a second type of antibiotic, streptomycin. So the way streptomycin works is that it actually binds into the ribosomes and it prevents the production of proteins that are essential for the survival of the bacteria. So again, streptomycin, the way it works is that it binds to the ribosomes and it prevents from the bacteria from making proteins that are essential for the survival of the bacteria. And the beautiful part about streptomycin is that just like penicillin, it only harms the bacteria without harming your cells. And the reason for that is because even though the cells in your body actually have ribosomes, the ribosomes that are found in your eukaryotic cells are different than the ribosomes that are found in the prokaryotic cells or the bacterial cells. Alright, so now let's start talking about the organelles. And the first organelle that we're going to be talking about is the cell membrane. The cell membrane, it's a barrier to the cell. Essentially, it's what keeps substances out of the cell and substances inside the cell. So the cell membrane is what we call a semi-permeable membrane. The reason why it's semi-permeable is because it allows certain things to go inside the cell and it keeps certain things outside the cell. So the cell membrane is made out of 2 layers of fat or 2 layers of phospholipids. And also within the membrane, you also have lots of proteins that are embedded within the membrane. And these proteins have a very important function. And we'll talk about the function in a few minutes. So a phospholipid is composed of a head, and then the head has a charge. Because it has a charge, it interacts with other molecules that have a charge. Molecules such as, for example, water. Water is a polar molecule because it has a charge. And so the head, because it interacts with water, it interacts with other molecules that have a charge, is what we call hydrophilic. Hydrophilic means that it's water-loving, if you will. Now the long tail of a phospholipid, because it's made out of a bunch of carbon to carbon to carbon to carbon bonds, and carbon, it's a neutral molecule. So that means that the tail of the phospholipid, it's what we call hydrophobic. So when you hear the word phobia, what does that mean? That means that you're afraid, right? So the tail of the phospholipid, it's afraid of water, so it points away from the water. And this is why you actually have 2 layers of fat, and the tails are pointing towards each other, and the heads are pointing towards the water. And because of this property, the cell membrane allows certain things to go in and certain things to stay outside. Okay, so this is something that I have already mentioned, but it's always important to hear it twice. So the cell membrane is semi-permeable. So essentially, it's made out of 2 layers of fat or phospholipid. And so the phospholipid consists of a head, and the head has a charge. And because it has a charge, it interacts with other molecules that have a charge, molecules such as water. So we call the head hydrophilic, which means it's water-loving. And then the long tail, because it's made out of a bunch of carbon to carbon bonds, and carbon is a neutral molecule, it doesn't like water. So we call that hydrophobic, because it doesn't like water. So it has a phobia for water, if you will. And so these 2 layers of fat are basically the tails are pointing towards each other, and the heads are pointing towards the water. Now, because of the very special properties of the phospholipid, certain things can go inside the cell, and certain other things are actually kept outside the cell. Now, simple diffusion is the movement of very small uncharged particles that can just basically go through the plasma membrane. And so those molecules go from an area where there's lots of them to an area where there's less of them. So essentially, they're going with gravity. And when it comes to simple diffusion, it doesn't require energy, and it doesn't require the assistance of proteins. So again, simple diffusion is the moving of very small molecules such as oxygen without the need of proteins and without the need of energy. So they go with gravity, if you will, from an area where there's lots of them to an area where there's less of them. Now, within the membrane, you also have proteins that are embedded in the membrane. And these proteins serves a very important function. So facilitated diffusion, it's the movement of large molecules, such as, for example, glucose. Glucose is a very large molecule, and it requires the assistance of proteins. Now, facilitated diffusion, these molecules are going from an area where there's lots of them to an area where there's less of them. And because they're going with gravity, it doesn't require energy. However, they do require the assistance of proteins. Okay, so one more time. Facilitated diffusion, it's the movement of large molecules such as glucose from an area where there's lots of them to an area where there's less of them. So they're going with gravity. It doesn't require energy. However, it does require the system of proteins. And the last type of movement that we have is called active transport. So in this case, in active transport, it requires the assistance of proteins. And because we're actually going against gravity, it requires the assistance of energy as well. So when you think about active transport, imagine going upstairs. So whenever you go upstairs, you're basically going against gravity. So gravity is pushing against you. So you're basically using energy. Again, active transport, it requires the assistance of proteins, and it also requires energy because you're going against the concentration gradient. You're actually going against gravity, if you will. All right, let's go back to antibiotics because that's what we were talking about. So antibiotics actually go inside the prokaryotic cells to the system of proteins. Now, let me ask you this question. Do you guys think that the bacteria is going to go down without a fight? No, the bacteria wants to survive. And so what happens is that the bacteria also has proteins that what they do is they're actually pushing out those antibiotics that you put into your body. And this is why it's extremely crucial and it's essential that whenever you go see a doctor and the doctor gives you antibiotics for 10 days, you should take them for 10 days. Don't make the mistake that many people do and, like say they take it for 2 days and they start to feel better and they're like, okay, I'm done. The reason why you should keep taking them up until, the 10 days is because that bacteria is going to be basically expelling or pushing out those antibiotics. And that's a good way how the bacteria become resistant to antibiotics. Essentially, what's going to happen is the bacteria is going to recognize that antibiotic and it's going to start producing more proteins. So that way, the next time you give those antibiotics, it's going to have enough proteins that is going to push all the antibiotic out. And in this case, the bacteria is going to become resistant.